question then is, why are there, let me take a guess here, approximately 150,000 papers since 1954, including, let me take a guess, 10,000 in the last year, with the title called The Isolation and Purification of Such and Such a Virus. And the 10,000, or I don't know what number I said, I've seen about 50 of them. The Isolation and Purification of the SARS-CoV-2 virus. And everybody who disputes me sends me these papers and says, you have to be wrong, because all the scientists in the world are saying they've isolated and purified this virus. So what I said up till now is how it should be done. And so why are they writing these papers that say the isolation and purification, and then they do the genome, and then they do the variants, and et cetera? What are they doing? This is the part you have to know, like making bread. So here's how they do it. This started in 1954 with a series of papers by a guy named John Franklin Enders, who was given the Nobel Prize in medicine, I think, for the isolation and purification of the measles virus and showing that the measles virus caused measles. This is where this started. Every single paper since 1954 has used the exact same technique that Enders inaugurated in his papers in 1954. And the reason this happened is because when they invented the electron microscope, which is the only way you can see a particle the size of a virus, in around 1935, they spent 20 years looking for virus from sick people and never found one. And so they came to a crossroads, as if to say, there's nothing to this. We were wrong. There isn't this pathogen that we can't see. We now can see it. Well, we could see it, but we don't find it. Now, the other important point to note here is one argument that you will hear is the reason you can't find viruses is because you can't do that. You can't find a particle that size. But what happened in 1930 was they took bacterial cultures, and they found that when you stressed and starved bacterial cultures, they broke down into these particles called bacteriophages, bacteria eaters, which are this identical size, genome, and structure of a virus. And they could find them in any bacterial culture. All you have to do is starve and poison a bacterial culture, and it coalesces down into this geometric form called a bacteriophage, and then they isolated and purified it. So it's not a technical problem. They could do this in the 30s. They could do it with so-called giant viruses, which are like spores in sea algae. And my friend, Stefan Lanka, was the first to find, by doing those exact steps, a virus in the sea, in the ocean. So it's not a technical problem of finding them. They just weren't there. And so they probably had a meeting and thought we should all be plumbers and carpenters because there's nothing to this. And Enders saved the day. So here's what he did. He took one person, one child with measles, not 100, not 500, not 1,000. One child took some of their bronchial secretions. And remember, the game we're playing is find the protein coat with genetic interior, right? That's the game. So the last thing you want to do is confuse the issue by mixing the lung fluid with some other genetic material, right? Then you're screwed. So what does he do? He puts the swab in milk, which is a rich source of genetic material. And I have the paper here that he wrote on this. Then what did he do? He took the swab with this fluid from the person with measles, put it in milk, 2 milliliters of milk, and then he mixed it with penicillin and streptomycin, 2 antibiotics. Then he mixed it with, quote, bovine amniotic fluid, 90%, a rich source of genetic material and proteins and particles, beef embryo extract, horse serum, antibiotics, phenol red, and soybean trypsin inhibitor. And then he inoculated that, smeared that onto monkey kidney cells. And then the mixture broke down. That's called a cytopathic effect, not the mixture, sorry. You have to be very clear. The vero cells, the monkey kidney cells, disintegrated. He also took away all the nutrients from the medium, or most of them. It's called minimal nutrient medium. So in other words, you have kidney cells and you feed them good kidney cell food. And then you start the experiment and you take away 90% of their nutrients and you add nephrotoxic, kidney toxic antibiotics to the mix. Now we use gentamicin and amphotericin, otherwise known as amphotericin, because it destroys kidney tissue. Then we add horse serum, embryonic fluid, fetal cells, etc., and then we get a cytopathic effect. In other words, the monkey kidney tissue breaks down. And that's called the isolation of the measles virus.
Oh my gosh.
The fact that the monkey kidney cell broke down, that's called proof that viruses cause disease. The fact that it breaks down into thousands and millions of little particles is called the isolation of the virus, and every subsequent genome experiment, PCR test, variant is a analysis of the fluid from the broken down tissue. Is that clear? Now, interestingly, Enders, in his original paper, and I'm going to quote it for you, says, quote, I'll interpret this. A second agent was obtained from an uninoculated culture of monkey kidney cells. The cytopathic changes it induced in the unstained preparation could not be distinguished with confidence from the viruses isolated from measles. Here's what happened. He said, well, I better do a control. So he started not with something from anybody with measles. He just did the milk, the antibiotics, the starving, the bovine this, smeared it on kidney cells, and he got the identical result. Think about that. In other words, it had nothing to do with anything from somebody with measles. And then in the follow-up paper, he says, quote, there is potential risk of employing cultures of primate cells for the production of vaccines composed of attenuated virus, since the presence of other agents possibly latent in primate tissue cannot be excluded by any known method. We have no way, he just said, of saying that these particles we're seeing came from the person with measles or from breaking down of the monkey kidney cells. We have no way of telling the difference. By the way, a live viral vaccine is that steps, right? They take the results of that culture, that's called a viral culture, and they inoculate that, and that's called a live viral vaccine. I don't think everybody got that. So you take the measles person and you mix it with milk. It's not exactly the same. You mix it with antibiotics, you starve the tissue, you put horse serum, fetal embryonic fluid, smear that on monkey kidney cells, it breaks down. You call that a virus, you call that isolation, you put that in a vial, and then you give somebody a measles shot. And an attenuated virus is they take some of the proteins out of that viral culture and not the whole thing, and that's called an attenuated viral vaccine. All vaccines are made in this identical way. All of the viral isolation studies, all of the DNA analysis, all of the genome analysis, all of the variants were discovered in exactly this way. Now, that way is a fraud.